First, we start, yesterday I spoke a lot about the flexibility, conformational variability of proteins. Proteins are drug targets, almost all drugs target proteins, and at least the approved ones. And we have, I'm not going to go into that anymore, but the whole idea, I think the concept is that we have moving targets. Our targets are moving. They keep changing their structure. They keep exposing a given region or not. And in all the drugability design, when we are designing drugs, we need to make sure that in our simulations, the targets are flexible enough. They are conformationally flexible. And within the context, I mentioned how you can generate an ensemble of conformers yesterday, an ensemble of conformers which you use for docking simulations, you use as input for any docking software. But an alternative approach, another approach, a little bit more time-consuming, but at the same time more rigorous, is drugability assessment with the help of MD simulations. So that's something where you now use also your MD simulation skills, your NAMD package. And we'll talk about that. What does it mean, drugability? It means the fact that the protein is druggable. What is druggable? You can bind a drug. So not all proteins, not all targets are druggable, and we'll talk about that. So we start with the druggability part, and in ProD there is a module, drug-GUI. It comes from drug, and GUI is the graphical user interface, which will allow you to do this type of analysis. Okay? And Indira will do the hands-on application for that. Now, I would like to give you some feedback, some motivation. This is a classical paper by Andrew Hopkins. It has been published in 2002, but there have been, since then, the new data just simply further confirmed, consolidated the initial findings. So he was then at Pfizer, and he was really struck with the fact that many targets which they want to design a drug for would not bind a drug or would not result in effective disease control. So, and usually we don't know about what we don't know. We don't know about those failures, because what's published is always the success stories. But when he had really data, a lot of data on what failed, and then he analyzed statistically, and he came up with those estimates. And since then, again, there are new versions that have been published. There is even a database now of druggable genes. And it is simply confirming these original results. The human genome, it has been shrinking, Now the consensus is 24,000 genes. That's what we have. But much more proteins, because there are different gene recombinations, Now, of those 20,000 genes, 3,000 are druggable. So this is what has been extrapolated based on the genes that have been really targeted and that are estimated to be targetable. And on the other hand, not all genes are related to diseases. An estimate of what, the fraction of genes that are related to diseases, disease-causing, and sometimes you can say disease-related, because they may be also preventing. In this case, you want to enhance their activities by agonists, right? So drugs are not always inhibitors. In some cases, they are agonists. They are not antagonists. So the disease-related genes is about 3,000. So what is interesting is that in that two subset, the intercept is of interest, right? The intersection is of interest. So we have practically, let's say, 1,000 proteins, drug targets, that are disease-related and druggable, okay? And this is good news in a sense, because it's not a big, infinitely large set. We can deal with it, especially with computers. And that is what the first thing, there is, drug discovery is a very, very time-consuming process. And they call this drug attrition effect. At later stages of clinical trials, you start dropping many, many drug candidates, mainly because they don't fulfill the what is called ADMET requirements: absorption, excretion, toxicity, etc., metabolism. So we have, but to avoid that, to avoid all these things, you need really to make sure that in the first place, what you are working for, what you are aiming at, is a protein that is druggable. Okay, these were a few concrete numbers then that he had. As you know, GPCRs are G protein coupled receptors. This is a very, very important group of drug targets. Kinases, obviously, but not phosphatases. Kinases are good drug targets, but not phosphatases. What is the difference? Do you have any insight on that? It's all about their structure, actually. Kinases are usually formed by multiple domains. They present a cleft in between, opening, closing. Phosphatases have flat surfaces. So kinases easily bind and stabilize drugs. Phosphatases are very slippery. They wouldn't. Okay. Now a few numbers again. And these slides will be accessible online, like those I presented yesterday. Now, this is a new number. Only 2% of human proteins currently are known to be targeted by approved drugs. Okay? Usually it's FDA approved, but we are now considering everything, worldwide. 2% of human proteins. It's really a small fraction, right? And from the previous slide, it was the ratio was about 15%, but a more conservative range is 10 to 15% of the proteins are disease modifying. 10 to 15% are druggable. So these three are based on what I've just shown, actually. And 5% are both druggable and disease modifying, and this is our, goal to really mine this set and design new drugs. So you can see that there is 2% already targeted, and there is probably another 3% we can easily. It's like a low-hanging fruit. They are waiting there. Okay. And so again, some background, general background. Among the 1362, and I think we have more updated numbers. I should have corrected that. But let's say 1,500 human proteins that are used as drug targets. Okay, and you can include a few more mammalian. The distribution here, published here in 2013, so it's more or less updated. They considered 4 mammalian species, mice, and I don't remember which ones exactly, including humans. And they just examined what are the distribution of those drugs, of those proteins. Obviously, a large fraction of them are membrane proteins, for obvious reasons. Why membrane proteins are better targets? Because they are on the membrane of the cells. They are easily accessible. You don't need to penetrate into the cell, right? But still, we have quite a large fraction of cytoplasmic targets, those in organelles, etc. So this is a distribution. And the interesting news is that, okay, membrane proteins have been targeted, and these are the class of proteins for which we have the least information. These are the hardest to resolve structurally, because they are stable in this lipid environment. So it's really hard to crystallize them unless you design some, the environment in liposomes, something. And also, most of them are really big. They are multimeric, and so they are really hard to structurally characterize, and they have been important drug targets looks like it's a good field to investigate, isn't it? And not surprisingly, NIH is allocating really tons of funds to explore, to understand better the dynamics of membrane proteins. And yet not so, uncharted territory, I would say, which has a lot of potential. Okay, in the absence of knowledge, any drug discovery process, they will tell you there are essentially at the molecular level, there are two approaches. One is ligand-based, the other is target-based. Okay? The ligand-based approach, ligand-based strategy, this is usually the recipe, the way you do it. You start with, usually, you start with some data here. Some data from, for example, from the literature, from our experimental collaborators. Now, you know that for a given disease, you have 20, 30 compounds that have shown some, desirable activity. They are not perfect, but they are a good starting point. Okay? Usually, that's the starting point. Then you generate what's called a pharmacophore model, and we'll get to that, using quantitative structure activity relationship tools, methods. A pharmacophore model is a model that has pharmacological properties. So it's, unfortunately, this is an overview, and then I will explain what a pharmacophore model is, okay? Now you have a model for what a desirable drug is supposed to be just from the analysis of those compounds. In your model, you will know that, okay, I need to have an aromatic group. I need to have an electron donor here. I need to have a long aliphatic chain over here, etc. So your pharmacophore model is not only the type of species, type of chemical species, but also their organization in space. Okay, there's 3D. They call them 2D fingerprint or 3D fingerprint. Each compound has a fingerprint. Okay? So now, based on the commonalities, you got your pharmacophore model. Now, you use this model to mine existing databases of compounds. This is called virtual screening. You do it on a computer. Of course, experimentalists also do this type of screening. Just starting from scratch, high throughput screening is a widely used and very ineffective method in experiments because you get lots of data and you don't know what to do with that. So instead, virtual screening is much more efficient. At least you don't spend so much time and money. Now, from your, now this is like a filter. You have a model, right? You have a pharmacophore model, and you would like to see in the database that you are screening the compounds that match your model. That's the whole thing. And then you extract from here new compounds. You do some standard, there are well-defined rules for selecting drug-like compounds. And then the next step is that you go to your experimental collaborators and you say, okay, based on what the initial raw data, now I screened, I mined large databases, and now I got new hits based on my virtual screening, and this is in vitro testing. These are relatively easy tests to refine the model, and then this is a little bit like an iterative scheme. I would even say based on that, you can go all the way to the original training set, which you can improve, and etc. So that's the strategy. And one thing you may have noticed in all that, there is no mention of the target. And guess what? Right now, the large majority of FDA-approved drugs are discovered this way, like trial and error. Okay? They are not designed rationally. Rational drug design requires knowledge of the structure of the target. Here, they don't even know what the target is. But this is just, based on, this is a trick used everywhere. You just use some existing data, you do computer screening, virtual screening, etc. And when I say screening, those databases of compounds, the libraries of compounds, they contain usually, let's say, 1 million different compounds. We're talking about that. And you can, select the best and then let them go through some filters. So this is like a funnel that gradually decreases to end up with a small set that will be tested experimentally. So it's known, a funnel-like description really works well for that. All right. Now rational design. We would like to design things rationally, right? Even though, and it looks like, rational design is important if you are really going to do something de novo, totally new from scratch. The others are kind of using existing knowledge and mimicking and trying to find more potent drugs. This is from scratch in a sense. This guy is, who is this protein? HIV protease, okay? Very important for HIV replication. So this is one of the drugs where some rational design really worked. And ideally, what you're doing is, if you analyze this protein by A&M, if you give its a code, PDB code to ProD, for example, and we have another server, A&M server, that's even easier for experimentalists. What it's going to tell you is that this has a tendency to move up and down like that. It has two monomers. And it turns out that, the effective drugs are actually, when the flaps here open up, they enter. So think if it were rigid, you would never have access to this region by the drug. The fact that it opens up and lets the drug in is critically important. So now the thing is, you have a target molecule, you try to see what are the critical regions, and this type of interfacial mobility is very important, and that's actually where the most effective HIV protease drugs bind. Okay. Of course, visual inspection, it's not a good thing, but you need to do that. There are things that robots can't do. You need to have some kind of intuition and start with some possible reasonable places for docking. This would be, but also you can do docking from scratch. There is a software called AutoDock where you don't know anything and just you search all the surface. However, again, flexibility is extremely important. And this is an example of rational design. So when I say first inspection, one thing you want to do is whether you have your candidate target, drug target, candidate drug target, if you like. Is it druggable or not? Suppose you have this protein now. This is the target, okay? And this is a classical surface representation. This type of representation, the blue regions would be positively charged, negatively charged, red, and white would be hydrophobic, neutral. Now, if you look at this, can you tell me, do you think that it's druggable or not? You can, actually. So what you look for, what do you look for when you just examine a protein structure and you want to see whether you can bind something? What? Pocket. A pocket, that's exactly true. You look for a pocket that would be really binding a small molecule. And preferably you want this pocket to be hydrophobic. I tell you, this class is really good. A hydrophobic packet, because the hydrophobic packet is not happy being exposed to salt. It wants to have a partner to limit the access to the. So is there a pocket here? And even a hydrophobic packet? Yes, where is it?
Below the two blue.
Over there?
Right there.
Okay, I'm just trying. This is it. You got it. Now you know exactly what to do with this protein. This is the drug indeed, and see, this is where it binds. You see the difference? It just inserts there. This is the real FDA-approved drug for that protein. It's a leukocyte glycoprotein, very important for adhesion, and without drug and with drug. So this seems to be contradicting everything that I said. It doesn't need to be opening up, or it has already this pocket ready to bind a drug, right? Well, there are some good, this is a good behaving protein that is already ready to bind a drug. So in some cases, this is really easy, but in many, many other cases, and that is what actually the results are, in the simulations, we've found that actually you could have an even higher affinity drug binding than what is best known. You know, the affinity of drugs, the binding affinity of proteins to the affinity of proteins to bind drugs, or the protein-drug binding affinity. It's usually measured in concentration of the drug. Nanomolar is really good. You have a very small amount of drug in your system, small molecule, and it still binds. Okay, this is the binding concentration. And we have, this one is 18.3 nanomolar, which is, this is experimentally detected quantity. When we do our, we published our analysis, we saw that actually we could even have a better binding affinity. This is not the best structure. You can even perhaps optimize further the interaction. But you know, already this amount of binding affinity is good enough for those purposes. Okay, what are durability simulations now? Now we have already learned two major things. Shape complementarity is very important in binding. And also, in addition to shape, you want to know some information about the shape chemistry, right? So many software, indeed, when they score the interactions, they have two scores, one for geometry, shape, and the other for the energetics. And they usually pay attention mostly to electrostatics because this is the strongest. Electrostatics is also a long range. It will help attract the ligand. Yes?
In previous aspect, how do you calculate the nanomolar of this simulation?
Yes.
How do you extract it?
I'll get there. This is the topic of my lecture. Okay. This, you calculate doing durability simulations. Okay. Now, what's durability simulation? It's very much inspired actually by what experimentalists do. We are mimicking what they are doing in the lab. We're just mimicking that in silico. So, for example, what NMR experimentalists do, they resolve the structure in the presence of different small molecules, and then they try to see the structure, so it is in solution, and you have many small molecules, and you even screen, you know, a library of small compounds, and you can identify from chemical shifts where this small molecule is binding, and then you have, and you can also identify, know whether it's binding where, you know, strongly or weakly, etc. So we're doing practically the same thing. And even X-ray crystallographers, you know, when they crystallize the protein, they soak it in the presence of small molecules, drug-like molecules, and then they look at the resolved structure where this molecule is located on the crystal structure. We're doing practically the same thing like NMR scientists because it's more in solution. And this is the physiological true environment. If you can see, the white surface is our target protein. We have our simulation box, usual simulation box for NAMD, for example. The only difference is that in the simulation box, in addition to water molecules, we put probes. What are the probes? Probes are isopropanol, acetamide, acetate, isopropylamine, and any other group small molecule you can think of. In our original study, we took this. Why did we select this? Because these turn out to be the functional groups or fragments that are most frequently observed in drugs, approved drugs. So for example, the isopropanol is observed in 57% of, what is that? I need to, now I can't even see. In 57% of known drugs, we have an isopropanol, at least one isopropanol group. Or acetamide, you know, we just selected these based on their frequency of occurrence. But you can increase this as much as you like. And then you start your simulations. And you know that this is going to be the binding site, right? But you don't, you want to see whether it is really going to be populated. And if it's going to be populated, what type of fragment will bind there? And then in your simulation, you gradually start to see that surface patch, which you might expect or not to be drug binding, is being occupied. And you do a probabilistic analysis. You try to see how often this region is really being occupied by a small molecule. So you have now, for example, when we applied, MDM2 was one of the proteins to which we applied. And you can, based on the affinity, on the frequency of occurrence of this binding, you can evaluate some binding affinity as you can estimate it. And then we color code the probes based on their affinity, how often they bind. This is a dynamic process. So to us, what matters is what fraction of time this site remains occupied. Okay? So the fraction of time gives you a probabilistic measure of the binding affinity, assuming that it's an ergodic process. So you have, the regions that show the highest affinity, we emphasize by just taking enlarging the size of the spheres there. Each sphere actually represents one probe, okay, molecule. And we just, to emphasize them, we just, the most likely site to be drug binding is now clearly seen. So Prudy has this module that would do this type of operations. And this is just showing an example, an application, a recent application, where you have a cytochrome C and a heme group. And in the simulations with a larger group of probes, you may notice that these are the probes all over the place. This is a sequence of snapshots, right, from the MD simulation. You notice this guy, this imidazole group actually binding the heme all the time. It just sticks there, likes it to be there. So, and that's what I mean by the pharmacophore model. Now we know that if we would like to design, if we would like to design a drug that binds this heme binding pocket of cytochrome C, we would like it to contain an imidazole group because imidazole likes to be there. So one part of it should contain an imidazole. And maybe next to it, we would like to have, I don't know, a negatively charged entity, etc. But this is all statistical analysis. Okay? And actually, well, these are, although I put there, this is from a paper that has been also recently accepted for publication in molecular pharmacology. So these methods are really very widely used for understanding the simulations are widely used, but the drugability simulations are just starting now to be more broadly used. Let's see. So again, back to what we're doing, the simulations that I'm talking about are short. This is good news. Even if you do a short run of 10 to 20 nanoseconds, you already get an idea of what the binding sites are. You don't want to do a wrong simulation of, let's say, 200 nanoseconds. Instead, 10 simulations of 20 nanoseconds are better. You want to ask something?
Yes. About that, because the off rates of these nanomolar affinity fellows are usually on the order of seconds, right?
Oh, that's, well, it's not actually. It's not, because you know, in this environment, it's much, much faster. It just in those simulations. So, you know, the experimentally observed off rates and those that we observe here temporarily are, I think, different things, different processes. The other one is really like a macroscopic view. Here we observe fast events on a very short time scale. So, so we do observe many on and off events, but it's not also binding tightly. Maybe in the experiments you have really something that binds tightly. So here it is just transiently, momentarily binding. This is what we observe. Okay? So based on this distribution, we call them hotspots. The regions that the ligand binds has, you know, showing a high affinity are hotspots. And then we calculate the achievable binding affinities. So the procedure, the recipe is, in the druggability simulations, you first heat it up, it is the annealing, and then you equilibrate it. So these are like the preparation phases. And then you conduct your simulation, druggability simulation of about 20 nanoseconds. Okay? And you can use any software. Now, your favorite software from now on is NAMD. So you can use it. Or you can use GROMACS. You can use whatever you like. All right. And again, a little bit more details, more quantitative details. Now, you got a series of snapshots, right? And you want, by the way, all these data Gs are rough estimates, okay? But at least to give you an idea. You want now you see in your simulation, suppose these are your isopropanol molecules. Isopropanol has CH3, CH3, CH2OH, okay? This is the formula. So you have two black and one red representing the isopropanol. And you would like, now, and then those red are simply the water molecules, simple representation. You would like now to assign, what is the binding affinity, right? Delta G, an estimate. So what you do is you use this formula. It's a very easy one. It's simply based on counts of number of occurrences. So how many, at a given region, how many, and I'm going to give you the formula. So suppose now you divide this region into grids, okay? And in the grids, you try to see here how many times within a given region you observe this isopropanol molecule. And you would like to normalize it with respect to a random distribution away from your probe molecule. It depends on the concentration, of course. So this is the observed concentration divided by the random concentration. So it gives you a measure of what is called enrichment. Yes?
The squares that you drew are different sizes.
They have the, well, yeah, we take care of those different sizes. I'm not going into those, of course. Number of cubes, there are such details. But the concept is to consider the difference between the random and the local number of binding events counted at different snapshots. So for example, when you do this type of calculation, if we go back, if you have a region where the number of bound states, observed bound states, is the double of what is expected, okay? And I over N0 is 2, right? If it's 2, you have ln 2. What's ln 2? Natural logarithm of 2. 0.69? Okay. What's RT? RT is the rate, like K, the gas constant or Boltzmann constant multiplied by the temperature. So R is, I could write these things, but R is 1.987 temp to the power minus 3 kcal per mole. And T is, let's say, at room temperature 300 Kelvin. You multiply them, the product is 0.6 kcal per mole. 0.6 kcal per mole multiplied by 0.69, it's about 0.4. So that's what this number tells us here, this number. So if you have a twofold enrichment compared to the random, your delta G would be of the order of, binding free energy would be about change in free energy that accommodates binding is about minus 0.416, okay? So this gives you an idea of the energetics. So when we have a series of hot spots here, if their energy is about, let's say, minus 2.5, it's a hot spot. But others, if it's only twofold enriched, it's a weak signal. You can call it a cold spot if you like. It's still a spot, though. All right. Any questions? No questions. Okay. So, the whole idea is to divide the space into grids. This grid trick is very useful. And then to count the occurrence of bound states in different grids as a function of time. You count how many times. And then you select those regions that show the lowest delta G. Low delta G means it's a good situation. No, the proteins, they don't like to have a high energy. All systems, natural systems, tend to lower the energy, right? So I'm going to skip that part. So suppose now you got an understanding of, okay, I'm going to have these probes. And then you may know also among the probes what are the different chemical entities that are most frequently binding in different regions. There are tricks, and I don't know if Indra will go into those. But suppose you have some probes that are overlapping. The energies are not additive. You cannot add them up. Okay? So we take account of this. And in the paper, those of you who are interested, the paper published 3 years ago outlines in detail, as well as our tutorials. So there are tricks to calculate, to avoid redundancy. You cannot add simply the energy of each probe to assign an affinity for a given site. But overall, after using these little details, technical details, you end up with solutions for your binding site. In this case, again, it's again MDM2P53. These are two proteins that interact. This is a harder problem because you want to block any protein-protein interaction. So it's not like binding a small molecule to a kinase. Now you want something that will be competitively binding instead of the partner. Okay. Now in this case, what is known from X-ray is this is the protein that binds. This was a very important finding, actually. This family of proteins that bind P53 and interfere with the interaction with MDM2. In our case, when we do this type of simulations, this is the type of structure, the probe distribution that we obtain. And we, this give us some idea. Let's see. Again, I think this picture is better. This is the protein, and this is the hotspots. And we can see that our hotspots actually more or less match the existing inhibitor. And that's what I mean by pharmacophore modeling. If you have those hotspots, now you know that you need a negatively charged group here, and you need an, aromatic group over there, etc. So you match your probe molecules, you build a pharmacophore model. The pharmacophore model is this shape of spheres, okay? You know their chemical identity, you know their position, the spheres. And you try to see whether it matches existing small molecules. And in this case, we know the answer, so it's just a verification that it gives reasonably good agreement. Okay. Now, an important concept in those simulations, what a big advantage of drugability simulations is the fact that you may not know at all the binding site, right? And what is even more important is the promiscuity of targets. What does it mean? A given protein may bind multiple drugs. It may have an active site, the catalytic site, but then there may be another hot spot, and this hot spot could be important for binding a substrate, right? And depending on, now if you bind something here and you block the catalytic activity, it's going to simultaneously inhibit the functionality of this molecule and simultaneously inhibit all its interactions. This is a drastic thing because the same protein may be involved in multiple pathways, and you want to be sometime more specific. So you want to alter one pathway while leaving the others untouched, right? So that's why it's important to also focus on recognition sites, hotspots for substrate binding. And we'll come to that later on. I will elaborate more on that. So this is, for example, another kinase where you have, from our simulations, we have identified multiple binding sites. And it turns out that one of them is the allosteric site for substrate binding. This one is for tubulin binding. And then there's a third site. Who knows what it is, but we are proposing it to be an important one. And we compare with experiments, etc. So there are many such examples. And I will just then let you play with this module. OK, the pharmacophore modeling, I promised that I would be giving you more details. So suppose this is, I should shift this slide up, I think. So suppose this is your starting set. You always have a good starting small set, of 20, 30 compounds. Now you superimpose those compounds, structurally align them. You structurally align them, and then you try to see what are, clustering is a very important thing. But clustering at the same time while aligning, clustering in 3D. And then you identify commonalities, you assign weights. Some positions may be really common to most of those drugs, others may not. In some cases, this may be 2 to 3 different classes you may want to separate and generate multiple models. But based on their superposition, now you have this. This is the pharmacophore model. This is what's called a pharmacophore model. It is just an image of spheres with, their spatial location, how far apart they are, roughly. You know what their composition is, hydrogen, acceptor, donor, etc. And then the next step is, now you got a pharmacophore model. This is your thing to use filtering when you screen libraries of small compounds, virtual libraries. And you can screen approved drugs or libraries of small compounds that may be potentially used as drugs. When you screen approved drugs, if you find something that fits this model, this is your model. Now you are putting different compounds, right? And you are trying to see whether it fits it. These are different trials. And suppose you find one that really fits well, then, OK, this is a good candidate for in vitro testing. If this is one of the drugs that's already approved, we call this a repurposable drug. OK? You can repurpose the drug. You can use that drug for your now new purpose. And finding repurposable drugs is very important. Why? Because you don't have to worry about all the clinical trials approval process. It has already proven to be not toxic, right? So you save time and money. Now, I just slightly touched on this issue of modularity and promiscuity, and I want to elaborate a little bit more on that. What time is it? How are you doing? Are you tired? Did I speak for an hour and a half? Oh, we started at 9. OK, I was thinking, what's happening? I didn't speak for only 45. That's good.
Imagine you only simulate 20 nanoseconds rather than 200. Did I miss why?
OK, why? Oh, because I didn't say why. And I should have. Thank you for asking. Now, what happens is when you have short simulations, as a physicist, you know very well this is at the minimum energy value. You are sitting at the minimum. In a short simulation, you sample the vicinity, OK? Now, if you have a longer simulation, instead of sampling the vicinity of your energy minimum, it's likely that it will probably cross a local barrier and jump into a new energy minimum. Okay? Then it will spend some time there and may get trapped there for a while. But the thing is, there are many, many local minima in the vicinity. And one given run will be just trapped there. Another one could be trapped elsewhere. So it is better to have a thorough examination of the closed neighborhood than having a totally statistically insignificant examination of longer. So that is all. It's for improving statistical accuracy. You want to get less results, but more accurate ones. Otherwise, they are not trustable, And this is a very, very well-established fact, in the literature. This is a recipe. You don't want to do long simulations. You want to use your time for multiple short simulations. Unfortunately, what Anton these days they want you to do is to take advantage of those supercomputer facilities and conduct very long runs, because this is what they think we have now accomplished, a simulation of microseconds. But then it's very risky. You do another run of the same duration, and you get different results. So I would always prefer to do shorter simulations and multiple runs. If you have infinitely larger capabilities, of course, do long simulations and multiple of them, but there is a trade-off. Yes, Simon?
Since you're doing short simulations, do you have to maybe bias the binding event so that you observe it in that short time window, or do they typically still bind it?
No, we don't want to bias, if you are, really very decided to target one specific site, you can do all, you can concentrate on that. But at the very beginning, at the early stage, you usually don't even know where to bind. You can explore the entire surface. So it is better to just start from scratch. But at a later stage, now you know the mutation site, for example. You know that there is a drug-resistant mutation at a given site. Then you may want to elaborate on a given. Yeah. Okay. Other important concepts. What a complicated sentence. Diversity and complexity of phenotypes arise from combinations of proteins and modular domains. Let me translate. The proteins themselves, by themselves, they are not so complicated. But now you have many combinations of different domains or proteins. And then these different combinations, and you know how things grow exponentially when you start to combine things. These combinations confer those specificities. Okay? So this is from a very nice review from the group of Wendell Lim at UCSF, where he has been emphasizing how if you have more modularity, with increasing modularity, the red diagram, you have a complexity of behavior. When you have instead a highly integrated small system, very integrated small system, then you can have, and this is what happens with small, easier organisms. You have less flexibility, less diversity in the type of interactions. So there are, I think it's very important to realize that when you have a given kinase, let's say, a given protein, you have the active site. You have the active site, which is here, usually between two domains. But then you may have another site here, because these are, kinases. A biochemist would say, oh, this is an enzyme. A biologist would say, oh, this is a signaling molecule. Okay? So kinase is a signaling. And the signaling would take place because of some conformational change elsewhere, where there is a substrate binding that senses a difference. And it may have some modularity, and this is like the SH2, SH3 domains for those of you who are familiar. So there are adapters, there is a scaffold region. So it gets increasingly more complex. And when we are trying to design drugs, it is very, as I mentioned, if we are targeting a specific biological function, then we need also to select not only the target, but also the site. Okay? So in the same protein, you can have many different sites. And depending on the site where you want to bind your drug, you will be interfering with different functions. This is for promiscuous proteins, of course. So this is a good example, again, from a recent review from a computational biologist, where Ruth Lusinov has considered GPCRs. Okay, GPCRs are, as I mentioned, they are really very important drug targets. And she's just emphasizing the fact that if you bind two different types of drugs, one is a normal agonist, an inhibitor, an enhancer, And then a biased, they call it a biased agonist because it ends up doing a different thing. They bind slightly different positions, but then they affect the interactions downstream with different substrates. One of them ends up affecting the arrestin-dependent pathway, and then the other one is the G protein-dependent pathway. So that's a very nice example of showing how you have the same protein and even two sites that are at close vicinity, but you may end up in different functionalities. So it is, this is called protein promiscuity. Many proteins are involved in, so this is a summary. Many proteins are involved in multiple pathways. And depending on the target surface region, or depending on the accessible structural change dynamics, because each region would be affecting the dynamics in a different way, you may affect in different ways the interactions with substrates, upstream or downstream. Okay? And this would result in different phenotypes. So that is a very important concept to bear in mind. Okay, let me try to conclude this part with a striking example, which I like. BCR-ABL tyrosine kinase. This is a very nice example of rational drug design, by the way. Prime example. Well, and this was discovered, and the discovery is a drug known as Glivec. You know, the more chemical name is imatinib, but Glivec is the brand name. Okay, when this appeared, even in Time magazine, that's what they said. Now there is new ammunition against the war, in the war against cancer. And these are the bullets, by the way, bullets. Yeah. So these are the drugs, okay? And this was a big, big discovery. Here is this is a kinase, and it has, again, two lobes, and this is the drug that binds right in the middle. It inserts there. Everything looked really good. The drug is, approved by Novartis. And then, what happens? A few years later, what happens? This is the usual thing that happens. Resistance. Who said that? So, okay, and sometimes they even call it multi-drug resistance because it's not resistance to one drug, but to multiple drugs. Drug resistance. And the drug resistance is so, funny. This is here where the drug sits, okay? This is the drug, the green one, and these are the coordinating amino acids. This is really rational. And then what happens is that in the wild type protein, there is a threonine that sits here nicely, and you can accommodate the drug. Okay, the drug can bind. But the protein, the kinase, instead made a small mutation and changed the threonine into an isoleucine. And guess what happens? Now it's protruding, right? And even this small change would not permit the drug, Glivec, to bind. So now we are dealing with a mutant. We are not dealing anymore with the wild type, and this mutant is drug resistant. So this has been a problem, and then there have been really lots of investments. This one is 200 nanomolar. It binds very nicely to the wild type. But when it comes to the mutant, the binding affinity is more than 10,000 nanomolar. Okay. Then the first thing you would do is the first strategy, at least in pharma, would be, okay, I know this drug approved in 2001, by the way, Novartis. Novartis is the first to further pursue that, of course. Their drug became now a little bit obsolete. And they try to use the same scaffold. What is using the same scaffold? For example, you see you have the same loop here. A little bit. So they just, think that, all right, this is the bad region. I need to make a change here so that I would avoid this adverse effect of this mutation, right? And indeed, they ended up approving this drug, designing a new drug that works, supposedly, and approved in 2003. And these are the two drugs, actually. This is one of them, and the other is the other, okay? Blue and purple. And here's threonine. But look at that. This threonine 315, the bad guy, it's still interacting with it, right? What type of design is this? And indeed, this, okay, they use the same scaffold. It didn't work. Then this one also got the same problem, although it was approved. Now another one, this time Bristol-Myers Squibb, they came up with dasatinib. And they are pretty much using the similar tricks, you know. They are also using the same, more or less the same scaffold, but they need also something called scaffold hopping. You know, you hop from, these are technical terminology. You just are taking a little bit a bolder phase and trying a different scaffold while maintaining some functional groups. And this is what is happening with different. To cut the long story short, none of them worked, okay? So the strategy of trying to optimize a drug that would effectively bind the kinase ATP binding site failed. Until these guys, this is again a joint study between Novartis and Harvard, until they came up with a really bright idea. And this is actually the second study, but the first was published somewhere else, but the second appeared now in Nature. It was accepted. Now, what are they doing? They're not targeting anymore this ATP binding site. They decided, no way, it's not going to work. But the same protein presents other sites, right? Now they are targeting an allosteric site. Allosteric is something that is far away from the active site, but an effect that would be also functionally relevant. And they still keep those inhibitors that bind the ATP binding site, like the Gleevec and other, dasatinib, etc. But now they have a new compound, GNF2, that binds a totally different position, a myristate binding site of the ABL kinase. And guess what? This, I'm going to show you here on the structure. And they detected the position with the help of NMR experiments. So this is where the Gleevec binds. Now when they do the NMR shift experiments, they know exactly which amino acids are being perturbed. And the new perturbation site is somewhere here. And this site is actually extremely important because this is the site that in the active state, this is only the kinase domain, there's an SH2 and SH3. This is called the post-translational modification or myristoylation site for the activation. So the chain, and it has to be myristoylated here. Now you are not letting it happen because you have bound here another drug that would obstruct the natural mechanism of activation. So the new drug that binds the myristoylation pocket has been to my knowledge, most recent knowledge, it is under clinical trial 2, and it may or may not be approved, but at least this is a breakthrough in the area. And actually some people in our university have been working on it too. Okay, I'd like to conclude that section with a big image, a big picture. So I mentioned that experimentally they do high throughput screening, but better than that is high content screening, where you have information on different simultaneously at the cellular level on the effect of a given drug. Of course, the challenge here is then to take those high content screening data, images at the cellular level or some tissue level, and then make sense of it. That's one thing many computational biologists are doing. Tim is an expert in this type of data analysis, for example. Tim gave you a lecture yesterday. And then from that, we now start to identify some commonalities, which features are giving the strongest signals. So we have this type of diagrams. So we can, let's say, we can build a network of proteins that are apparently, presumably involved in this, in the interaction, in the action of the different drugs. Having identified proteins, then you can focus on some functional sites or new sites. You can predict drugs for these targets. And also there is some, hopefully you could merge this with the cellular simulations like MCell. This is not yet done, but I think that's a missing piece that needs to be coupled. And then you have new predictions, new experimental tests, and this goes on and on, okay? So this is the big picture. And this is called quantitative systems pharmacology. It's a new area. It's not molecular pharmacology, quantitative molecular, it's quantitative systems. The whole idea is that you take the effect at the level of the entire cell, and you extract a network of proteins, and then in this network you identify some critical nodes which you would like to target, okay? And then you would know exactly the effect of obstructing that particular interaction because you know the network dynamics at the cellular level. Of course, we mine a drug bank and existing databases, and at the end, if everything goes well, you can go to clinical trials too. I would like to conclude this session by, again, a review. If you are interested in computational biology, this was written in 2009 by Janet Thornton. Janet Thornton is, I think, one of the pioneers in the field. She is #1 computational biology person in Europe. I'm not exaggerating. She's leading EBI, European Bioinformatics Institute in Cambridge. Well, she just stepped down a few months ago, but she had been leading it for quite some time. And she wrote this, you know, I would say not a review, an opinion article, summarizing what the role is for computational biology in the coming decade. And many things that she wrote there are already coming true. And you know, in your free time, leisurely, you may want to check that. Of course, data analysis is an important area. We take advantage of all accumulating data. And sequence-based methods, you know, Xian mentioned them. I will talk about them later on. Obviously, structure-based methods. Flexibility. I talked about that a lot, conformational split. These pathways or promiscuity concepts are, so she's just elaborating on really what are the directions, future directions, and how computational biologists will be sitting at the driver's seat, not in the back seat, okay? So that concludes the first part of my lecture today. And it's a good time to give a break. Is it? And then we'll just continue in 5-10 minutes. Be back in 10. Yes, of course. Sorry, I should have.
The resistance finite function. I thought active sites don't usually do that often. 2 years seems to be a very short time. And another related question is, when we talk.
What is the question? What's the first question?
Why does it mutate so fast? It's active site, right?
Yeah, but it's not anymore. It's not, you have already interfered with it, right? You're binding where the systems, the proteins have this tendency to restore function. You know, in the greater scheme, they call it homeostasis. But for proteins, now there is this word proteostasis. They restore their functionality. They have this evolutionary tendency.
You know, related to this question, if we compare, you know, person to person to look at this kinases, kind of homology, or you know, how conserved these residues are, we could avoid design drugs targeting, you know, that residues are more mutatable.
Yeah, yeah, that's a good point. Of course, you know, you may need to think of all possible scenarios when you design a drug. But it's really so hard to design something that works. So when something works, people don't even want to further elaborate. We got it, they say. But then after a couple of years, you have those drug resistance.
Yeah,
And in my next lecture, I'm going to talk more about that. Yes?
What's the structure of the chimeric protein such that you can bind the myristate binding site on ABL and then make the whole complex?
These are not other proteins. This is the same protein, another domain. So this kinase is composed of 4 domains. There's a N lobe, C lobe, SH2 domain, SH3 domain. So at the end of the SH3 domain, there's an N terminus that gets myristoylated and binds that exactly the same region to get the protein activated. Okay? Now, the N terminus cannot bind anymore this region in the myristoylated form because now it is already occupied by the GNF2, the new drug. So that.
So the normal ABL protein doesn't do that.
The normal ABL protein, to get activated, it's going to have this post-translational modification at its, did I say N-terminus? It's the C-terminal region, I think. Yeah, SH2 is the C-terminal region. So this region in the normal protein will get this post-translational modification, bind this, methylation group, and then it will interact with that exactly the site that I showed to get the kinase activated. There are many studies on that. If you are interested, I can send you one. Okay.
So in the BMD add-on, I saw that you have given only like 4 or 5 probe molecules. So if you want to change the probe molecule,
yeah.
we can change it?
Yeah, definitely. One bottleneck in those simulations is the force field. The force field is very well developed for amino acids in proteins. But when it comes to small molecules, which have different groups, different atoms, this is still a problem. Some of them, they don't have good parameters, but we just use whatever is available. We don't, we're not in the business of developing force fields. Others are doing that. And I think it's really boring. But as long as there are some parameters we can use, we can add any. For example, if we already know the binding site, and if we already have some, promising candidates with certain functionalities, we just dump them in with suitable force field parameters. Other questions? Okay, you can ask me questions later in 5 minutes.